So continuing on the topic of Evo-Devo, in Drosophila melanogaster, so the common fruit fly, the antennapedia gene is known to drive leg formation. So relocate the gene to a homobox region responsible for antennae development, and you get legs growing in the place of antennae, as you can see on the right here. So the antennapedia gene responsible for leg development. If you take that leg development gene out and you want to insert it into a Drosophila to make it have legs instead of antennae, all you have to do is insert that gene where the homobox gene responsible for antennae development is, and you're going to get legs growing in the place of the antennae. In spiders, the antennapedia gene represses leg formation, so silencing it results in spiders with more legs. These structures are produced in embryonic development, which operates within von Baer's laws. So a brief overview of these laws: their general characteristics develop before specialized ones. So for example, you get kind of like little limb nubs in embryos before you get the full kind of limb with digits forming. The form of an embryo does not converge with the forms of another species, it diverges from ancestral forms. So the end product of an embryo isn't going to follow the exact same path as other species, because they have different evolutionary trajectories. And the embryo of a higher animal does not resemble the embryo of a lower animal, except for in terms of retained structures from the higher animal's ancestral state. So, the similarities we see between the embryos of different species are the product of evolutionary conserved embryonic traits. For example, if you look at the image here, this is a human, I believe. You can see these structures here where they've kind of got the structures labeled the pharyngeal arches that look a bit like gills on the neck of the embryo. These pharyngeal arches are present both in fish embryos, and they're present in human embryos. And in fish, these have always given rise to the gills, but when the Sarcopterygian, the lobe-finned fish, moved to land, because they didn't need gills anymore, they adapted these pharyngeal arches to give rise to the structures of the neck that allowed them a more terrestrial kind of lifestyle. But it should be kind of noted that although both animals still have them, they're giving rise to different structures, but the pharyngeal arches are an ancestral state that modern tetrapods like humans, well, modern vertebrates like humans, have retained because it's a evolutionary conserved trait. So with regards to limb development, specific homobox genes activate the TBX4 to 5 gene that codes for limb specification. The limbs extend from limb buds in the embryo with two signaling centers controlling outgrowth. These signaling centers are the apical ectodermal ridge, which is in charge of proximal distal growth, so it controls the extension of the limb. If we cut this region off of one embryo and attach it to another, then the limb will continue to grow from this apical ectodermal ridge from where it had been previously growing before. So let's say in an embryo, this would be a normal kind of arm over here. Let's say that we kind of took development from the humerus in one embryo, so the apical ectodermal ridge is extended enough to develop the humerus. Then we cut that apical ectodermal ridge off of that embryo, and we stuck the apical ectodermal ridge onto this spot of the other embryo, the original apical ridge from this embryo is still going to continue to grow, so you're going to get the regular humerus, tibia, fibia, and then the digits of the hand growing. But in the apical ridge that we transplanted, it's going to continue growing from wherever it was at in the original embryo that we've taken it out of, and you're going to get full development of another kind of tibia, fibia, and the digits of the hand. And then the other signaling center is the zone of polarizing activity, which is in charge of anterior to posterior growth, so kind of the thickening of the limb rather than the extension of the limb. And that the zone of polarizing activity is responsible for tissue development of the limb. So something quite interesting, the sonic hedgehog gene is responsible for the zone of polarizing activity, and specifically the development of the digits, and it's regulated by several Hox genes. Changes to these digits tend to be a product of gene regulation to an extent. Whales have lost their hind limbs through evolution, but retain the structures that give rise to those limbs, which I'm sure that you've seen in if you've ever seen a whale skeleton, they retain kind of some structures of the pelvis, although they've fully lost their limbs. They've basically lost their hind limbs because they've shut off the Hox genes responsible for the limb development, so you're not getting this apical ectodermal ridge it's lengthening the limb to produce kind of back legs, because they have a tail instead, because obviously they're swimming. Disconnection of the Hox and TBX networks has allowed snakes to shut off limb development. So that's how they kind of went from an ancestral kind of tetrapod lizardy kind of state, like other squamates, like the lizards, and they lost their legs and they kind of become snakes. Gene duplication allows for modifications to be made to the new gene, allowing it to take on a new function, while the original gene retains its function. So this is a different kind of... I think I'll do gene duplication as a separate video, because it's not particularly related to the Homeobox gene stuff, and then that'll be kind of the last bit of the genetic side, and then we'll just have a look at parental care if I have time. So that's it for that one.